Welcome back to the Deep Dive. It is great to have you with us.
Great to be here.
Today we are tackling a subject that is, I think, simultaneously the most ordinary thing in the world and, frankly, the most impossible thing in the world. We are talking about the interface.
The interface.
Yeah, the biological machinery that lets you actually experience reality. I'm talking about how we see and how we hear. We open our eyes, we listen to something like this, and we just assume it's, the default settings. But when you actually look at the construction side, how an embryo builds these organs, it just feels like it shouldn't work.
It really is a high wire act. I think that's the perfect way to frame it. We aren't just talking about anatomy today. We are talking about a very high stakes architectural project where timing is everything.
Everything. And we are drawing from some heavy hitters for this deep dive. Langman's, Moore's clinically oriented embryology, and of course, Singh. And the picture they paint is of a process that is just incredibly aggressive and fast.
Fast is an understatement. I think the timeline is the first thing that really shocks people.
Let's start there. Because when I was reading the notes, I had to double-check the dates. We tend to think of sensory organs, eyes, ears as, like, finishing touches. You build the body, then you add the accessories. But that's not the case at all, is it?
Not at all. The blueprints for your eyes and your ears are being rolled out when the embryo is barely the size of a peppercorn. We are talking about day 22.
Day 22, that's the beginning of the 4th week. I mean, most people don't even know they are pregnant at day 22.
Exactly. And yet, biologically, the train has already left the station. At day 22, the neural tube, which is going to be the brain and spinal cord, is essentially just zipping itself up. It's still open at both ends.
Right.
But right there, in what will become the forebrain, you can already see the optic grooves. And on the side of the future hindbrain, you see the otic play codes.
So before the heart has fully figured out its plumbing, before there are even little limb buds, the brain is already trying to build a camera and a microphone.
Yeah. It prioritizes the interface. It's absolutely rushing to get the sensors online. But, and this is really the core theme of today's episode, the brain cannot do it alone.
And this brings us to that concept that Langman and Singh really hammer home. Reciprocal induction.
Yes. If you take one thing away from this deep dive, let it be that. Reciprocal induction.
It sounds like a legal term. Or maybe some kind of physics principle.
It sounds a little dry, I know, but it's actually incredibly dramatic. It's essentially a biological handshake. Or a conversation.
A conversation between tissues.
Exactly. You see, we tend to think that an organ just grows out of one place, like a branch growing out of a tree trunk. But complex sensory organs like the eye or the ear are never built by a single tissue in isolation. They are built by a committee.
A committee of tissues that normally don't even talk to each other.
That's right. You have the neural tissue, the brain, and you have the surface ectoderm, the future skin. Usually these two just stay in their own lanes.
Yeah.
But to make an eye, they have to meet. They have to physically touch. The brain reaches out and says, hey, I am going to build a retina here. And the skin has to answer, okay, if you do that, then I will build a lens.
So it's a dependency. One can't do it without the other.
It is total codependency. If the brain reaches out but the skin doesn't answer, no eye. If the skin is ready but the brain doesn't reach out, no eye. It's a choreographed dance where a single missed step means the organ simply does not form.
Which explains why things can go so wrong, which I definitely want to get to. But let's start with the eye, because the way the eye forms, it's kind of aggressive.
It is. It's an invasion, really.
So paint the picture for us. We are at day 22. We have the forebrain, the part we call the diencephalon. What happens?
Okay, so imagine the neural tube, the future brain, is a long, soft cylinder. On day 22, in the head region, you get these two shallow grooves appearing on the sides. These are the optic grooves.
And these aren't separate structures attached to the brain. They are the brain, right?
Correct. They are the brain wall itself. As the neural tube closes, these grooves deepen and they start to bulge outward. They become what we call the optic vesicles.
Like blowing two small bubbles out the side of a bigger one.
That's a perfect image for it. Those outpocketings are your eyes starting to form.
This is a distinction I think a lot of people might miss. The retina, the part of the eye that actually sees, is technically brain tissue.
It is direct neuroectoderm. It is an extension of the central nervous system. It's not like the brain. It is the brain, just extruded outward.
So that's why the optic nerve is a cranial nerve. It's not a peripheral nerve like one in your arm.
Exactly. It's a hardline cable straight into the mainframe.
Okay, so we have these optic vesicles, these bubbles of brain, and they're pushing outward through the mesenchyme, the sort of filler tissue. Where are they going?
They are pushing toward the surface. toward the surface ectoderm, the skin. And this is where that handshake happens.
The contact.
The contact. The optic vesicle grows laterally until it physically bumps into the surface ectoderm. And that contact is the moment of induction.
And the conversation begins.
Immediately. The brain tissue releases chemical signals, things like BMPs and FGFs, which are basically molecular text messages that tell that specific patch of skin, you are not going to be skin anymore. You need to become a lens.
And the skin reacts.
Instantly, the cells get taller, the tissue thickens, and we call this thickened patch the lens placode.
The analogy that came to mind when I was reading the sources was pressing your finger into a soft balloon or maybe a piece of dough.
That is the perfect visual because of what happens next.
The geometry gets really wild here. So the brain pushed out, that's the finger.
Yeah.
And the skin thickened, the placode. But now the skin pushes back.
Yes. This is the reciprocal part. The lens placode invaginates, it sinks inward back toward the brain. It forms a pit, the lens pit.
Okay.
And eventually the edges of that pit come together and they pinch off completely.
So we've basically captured a bubble of skin inside the head.
That's exactly what it is. It becomes the lens vesicle, a hollow ball of surface cells now floating inside the head, completely detached from the outside world.
Okay, but what happened to the brain bubble, the optic vesicle, while this skin bubble was sinking into it?
Go back to your finger and the balloon analogy. If you push your thumb really deep into a soft ball, what happens to the ball?
It collapses.
Yeah.
It turns into a cup shape.
Precisely. The optic vesicle collapses inward to form the optic cup. And this is critical because that cup is now double-walled. You have pushed one wall against the other.
And this double-walled cup becomes the retina.
It becomes the blueprint for the entire back of the eye. And because it's a folded cup, you have two layers. The outer layer, the back of the cup, becomes the pigmented retinal epithelium.
Those are the dark cells, right?
Yes. Their job is to absorb any stray light so it doesn't bounce around inside your eye and blur the image. They are the blackout curtains of the eye.
And the inner layer, the part of the cup that got pushed in.
That becomes the neural retina. That is the complex part. That is where the rods and cones, the bipolar cells, the ganglion cells, that's where they all develop. That is the film in the camera.
Now here's a detail from Moore that I found fascinating and honestly a little unsettling. There is a space between these two layers, right? The intraretinal space.
Yes. Originally, that was the inside of the brain bubble, the vesicle. When the cup folds, that space gets squished. The two layers, the pigment layer and the neural layer, are just pressed against each other.
But do they fuse? Like, do they weld together?
No. And that is the kicker. They do not biologically fuse. There are no cellular bridges, no desmosomes locking them together. They were just held together by pressure and fluid dynamics.
That seems like a massive design flaw.
You could certainly argue that, because that intraretinal space is a lifelong weak point. In an embryo, it disappears. But in an adult, if you take a severe blow to the head, or sometimes just spontaneously as you age, that space can snap back open.
And that is a retinal detachment.
That is exactly what a retinal detachment is. It isn't a tear in the tissue, usually. It's the neural retina peeling away from the pigment layer, reopening that embryonic space from day 28.
Ooh.
It's the eye reverting to its developmental geometry.
That is wild. So a boxer getting hit in the face today is exploiting a gap left over from the 4th week of their own life.
It really highlights how our developmental history is written right into our anatomy. We are walking around with these embryonic fault lines inside of us.
Okay, so we have the cup, we have the lens sinking in, but we have been talking about the eye as a singular object. But we have two. And looking at the genetics in the notes, that wasn't a guarantee.
No, it was not. And this brings us to the molecular architects. The genetics here are fascinating, and they explain some of the most tragic birth defects we see.
The source material identifies a master gene,
PX6.
PX6, the boss.
The boss. PX6 is the master regulator for eye development. If you delete PX6, you get no eyes. Period. But here's the trick. Initially, PX6 is expressed in a single band, a stripe, right across the front of the neural plate.
So technically, we start with a cyclops eye field.
In a molecular sense, yes. We start with one continuous potential eye field running across the midline of the face.
How do we get two eyes? How do we split that field?
We need a wedge. We need a signal to come in and say, no eyes here in the center, please. Push them out to the sides. And that signal comes from a structure called the precordial plate, which sits right in the midline. It secretes a protein with, I have to say, the best name in all of biology.
Sonic hedgehog.
Sonic hedgehog. SHH.
I still can't believe that's the real scientific name.
It is, and it is incredibly potent. Sonic hedgehog basically acts as a suppressor. It suppresses PX6 activity right in the center of the face.
It creates a no-build zone.
Exactly. It splits that PX6 field into two lateral patches, so now you have a left eye field and a right eye field. And in the middle, where it cleared the space, it upregulates another gene called PX2.
And what does PX2 do?
It builds the optic stalks, the wiring, so you end up with this beautiful gradient, PX2 in the middle for the optic nerves, PX6 on the outsides for the eyes themselves.
But if that signal fails, if Sonic Hedgehog doesn't show up for work,
if that signal fails, and this can happen due to genetic mutations or even certain environmental toxins, the field never splits.
And that is where we get cyclopia.
Yes. Synophthalmia, where the eyes are fused in the middle, or true cyclopia, a single midline eye.
The notes mention a specific environmental trigger for this, something about corn lilies.
Right. This is a famous story in teratology. There is a plant called Veratrum californicum, the corn lily. It contains a toxin called cyclopamine.
Cyclopamine, that's aptly named.
It is. And if a sheep eats this plant specifically on day 14 of gestation, the toxin blocks the sonic hedgehog signaling pathway. The eye field doesn't split, and the lamb is born with a single eye.
It is terrifying how specific that is, just day 14.
It proves the mechanism. And in humans, this condition is usually associated with holoprosencephaly. That's a failure of the forebrain itself to divide into two hemispheres. Because remember, the eye is the brain. If the brain doesn't split, the eyes don't split.
The face predicts the brain.
Exactly. That is a clinical maxim. If a baby is born with eyes that are too close together, hypotelorism, it is a massive red flag that the midline of the brain might not have formed correctly.
It's tragic, but it really underscores the precision we're talking about. You need this molecular wedge to literally drive the eyes apart.
And once they are apart and you have that optic cup we talked about, you still have some plumbing issues to solve.
Right, the fissure. Because the optic cup isn't a perfect bowl, is it?
No. Imagine holding that tennis ball cup again. But now, imagine you need to run a blood supply into the center of it. The lens is growing like crazy. It needs nutrients. You can't just seal it off.
You need a slot, little access panel.
You need a slot. This is the choroidal fissure. It's a slit running along the underside of the optic stalk and the optic cup.
And what runs through it?
The hyaloid artery. It's a temporary artery that tucks inside the eye to feed the developing lens and the inner retina.
But looking at my own eyes in the mirror, I don't see a slit. I see a nice round pupil.
That is because around week 7, that fissure is supposed to fuse shut. It zips itself up. When the lips of that fissure meet and fuse, the mouth of the cup becomes a round pupil.
And if the zipper gets stuck?
That is a condition called a coloboma. If the zipper stops halfway, you are left with a gap.
And this gives that keyhole look, right?
Exactly. If you see a person with a pupil that looks like it's spilling downward into the iris, looking like an old-fashioned keyhole, that is an iris coloboma.
Is it dangerous?
It depends on how deep the failure goes. If it's just the iris, it might just be a cosmetic difference and some light sensitivity. But if that zipper stayed open all the way back to the optic nerve, well, that can cause serious vision loss. It's essentially a failure to close the construction hatch.
Speaking of construction scaffolding, let's talk about the lens again, because there is a paradox here that I absolutely love. The lens needs to be transparent.
Crystal clear.
But living cells need blood, and blood is red.
Yeah.
And cells have organelles, nuclei, mitochondria that scatter light. So how do you make a living window?
You have to create what I like to call transparent zombies.
Transparent zombies. Okay, I'm listening. Unpack that.
So go back to that lens vesicle, the bubble of skin. The cells at the back of the bubble start to elongate. They grow forward, filling the whole cavity. These become the primary lens fibers.
But they are still normal cells at this point, with a nucleus and everything.
Yes, and they are being fed by that hyaloid artery we just mentioned. But to become the lens we need, they have to commit a sort of partial suicide.
Explain partial suicide.
They systematically destroy their own nuclei. They break down their mitochondria. They strip away everything inside themselves that could possibly scatter a photon of light. They then fill their cytoplasm with special proteins called crystallins.
So they are technically alive, but stripped of all the normal machinery of life.
Exactly.
Yeah.
They can't divide. They can't repair themselves very well. They are just these perfectly ordered bags of crystal maintained by the cells around them.
And the blood supply, the hyaloid artery.
The blood supply has to go too. The hyaloid artery, once it has done its job of helping to grow the lens, receives a signal to die. It undergoes apoptosis-programmed cell death.
So the scaffolding is taken down before the building opens for business.
Exactly. By birth, the lens should be completely avascular. It has no blood supply. It just sits there, sipping nutrients from the aqueous humor fluid in the front of the eye.
But if that artery doesn't die,
You can get a persistent hyaloid artery. You can actually see a little worm-like vessel floating in the eye, blocking vision. But more commonly, if the lens fibers don't form right, or if an infection hits them while they're trying to become transparent,
they turn cloudy.
they turn opaque. This is a congenital cataract, and this is a good moment to bring in the villain of the first trimester, the rubella virus.
German measles.
Right. Rubella is a classic teratogen, a monster maker. It just loves rapidly dividing cells. And it creates what some texts call a Grecian profile, a viral attack.
Grecian profile. What does that mean in this context?
It means the virus hits whatever is highest on the construction list at that specific moment. It leaves this chronological timeline of damage.
So if a mother contracts rubella in, say, week 6,
in week 6, the lens fibers are forming. They are incredibly vulnerable. The virus infects them. They fail to become transparent. And the baby is born with cataracts, white pupils.
But if the infection happens later, say, week 9.
By week 9, the lens is mostly finished. It's pretty much sealed off, so the eyes might be fine. But the ear is now under heavy construction, specifically the organ of Corti, the hearing part. So a week 9 infection spares the eyes but can destroy hearing.
It's a really sobering thought. The virus doesn't choose a target. It just hits whatever window happens to be open at the time.
Exactly. Biology is all about timing.
Which is the perfect transition to our second sense, the ear. Because if the eye was the story of the brain pushing out, the ear seems to be the exact opposite.
It is the inverse. The inner ear, the labyrinth, starts as the skin sinking in.
So we are back to day 22.
Right.
Back to the surface ectoderm.
Right. On the side of the hindbrain, the rhombencephalon, you get another thickening, the otic placode. And just like the lens, this placode invaginates. It sinks in to form a pit, then it pinches off to form a bubble.
The otic vesicle.
Or the oticis. So now, just like with the lens, we have this little bubble of skin tissue trapped deep inside the head.
But this bubble has a much more complicated destiny than the lens. I mean, the lens just has to be a clear ball. This otic bubble has to become the most complex labyrinth in the body.
It has to do two completely different jobs: hearing and balance. And to do that, it has to change shape dramatically.
How does a simple bubble become the inner ear?
Well, it divides, it elongates, and it warps. The dorsal part, the top part, is destined to become the vestibular system. That's the utricle and the semicircular ducts.
That's the balance hardware.
Right. And the geometry here is just beautiful. Think about what the semicircular canals are. They are 3 loops oriented in the X, Y, and Z axis.
They detect rotation in 3D space.
Exactly. But how do you make a loop out of a bubble?
Yeah, how do you do that?
It's like sculpture. You start by growing a flattened disc extending out from the utricle. So imagine a little pancake sticking out of the bubble.
Okay, I have the pancake.
Now the cells in the center of the pancake receive a signal to die. Apoptosis again.
Apoptosis is really the sculptor of the embryo, isn't it? It's just everywhere.
It is. It carves by deletion. The center of the pancake dissolves away, leaving only the rim. And that rim is the semicircular canal.
That is incredibly elegant. You make the tube by deleting the middle.
And because there are 3 of these discs oriented at right angles to each other, you get the 3 canals that let you know if you are nodding yes, shaking your head no, or tilting it to the side.
Okay, so that's the top part, balance. What about the bottom part, the hearing part?
The ventral part of the otic vesicle. This grows into the saccule and the cochlear duct. And instead of making loops, this part decides to spiral.
The snail shell, the cochlea.
It grows in curls. Specifically, it makes about 2.5 to 2 3/4 turns.
That's specific.
Oh, very specific. If it stops short, say only 1.5 turns, you get a condition called Mondini dysplasia, and you can lose low-frequency hearing. The geometry dictates the function.
And inside this spiral tube,
the cells differentiate into the organ of Corti. These are the famous hair cells, the transducers. They're the things that turn physical vibration into electrical impulses that the brain understands as sound.
And again, I'm sure there are genes running the show here.
PX2 makes a comeback here to regulate the cochlea. And another gene, TBX1, is crucial for the cochlear duct. If you knock out TBX1, the cochlea fails to spiral properly.
But here's the thing that always confuses me about the ear. We have built the inner ear, the cochlea deep inside the skull. That's great. But sound comes from the outside.
Right.
We need a way to get the sound to that little bubble.
You have just hit on why the ear is so much more complicated embryologically than the eye. The eye is mostly one packaged neuroectoderm and surface ectoderm shaking hands. The ear is a scavenger hunt.
A scavenger hunt.
It is 3 separate components from 3 different origins that all have to find each other in the right place at the right time. We have done the inner ear, which is from the surface ectoderm. Now we need the middle ear, the amplifier.
And looking at the sources, the middle ear comes from the throat.
It sounds bizarre, but yes, it comes from the endoderm. Specifically, the first pharyngeal pouch.
Explain the pouch.
So, imagine you are inside the primitive throat. There is a recess, a pocket, that extends outwards toward where the ear is developing. This is the tubotympanic recess. It grows outward until it touches the location of the external ear canal.
So the cavern of the middle ear, the space where the bones sit, is actually a throat extension.
Yes, and that is why your ears pop when you swallow. The eustachian tube is the stalk of that original pouch. It still connects your middle ear to your throat.
And the membrane where this throat pocket meets the outside world.
That becomes the eardrum, the tympanic membrane. It is this perfect sandwich of all 3 germ layers, skin on the outside, ectoderm, fibrous tissue in the middle, mesoderm, and throat lining on the inside, endoderm.
Okay, so we have a cave. But a cave needs machinery. It needs those tiny bones, the malleus, angios, and stapes. The ossicles.
The ossicles, the smallest bones in the human body. And they don't come from the pouch. No, they don't come from the otic vesicle. They come from the pharyngeal arches.
Which are neural crest origin, right?
Mostly, yes. The first pharyngeal arch gives us the malleus, the hammer, and the incus, the anvil. The second pharyngeal arch gives us the stapes, the stirrup.
I love the evolutionary history here. This is just a spare parts project, isn't it?
It absolutely is. If you look at a reptile, these bones are part of the jaw joint. They're used for chewing. In mammals, evolution repurposed them. It migrated them upwards into the ear and turned them into a precision amplification system.
So we are literally hearing with our ancestors' jawbones.
Essentially, yes. But because they are separate pieces coming from different arches, things can go wrong during the assembly.
Like what?
Well, specifically with the stapes, it's supposed to sit in a little window called the oval window right against the inner ear fluid. But if the differentiation isn't perfect, the stapes can remain fused to the surrounding bone.
Stapes fixation.
Exactly. The bone is locked, it can't vibrate. So sound hits the eardrum, travels through the hammer and anvil, and then just hits a brick wall. The person has conductive hearing loss because the last bone in the chain is stuck.
The machinery is jammed before it was even used.
Jammed by a failure of the cartilage to differentiate properly.
And finally, we have the external ear, the part we can actually see, the pinna.
The collector. This is another migration story. The external ear forms from 6 little bumps called auricular hillocks.
Hillocks.
Little hills. 3 come from arch 1, 3 from arch 2. And they appear around the first pharyngeal cleft.
But where do they appear? Because looking at the diagrams in Langman, they're really low.
They appear on the neck.
On the neck.
Yes. Initially, your ears are very low. As the mandible, the jaw grows and the face develops, the ears are pushed upward and backward to their final position on the side of the head.
So the ears don't really move. It's more that the face grows out from under them.
Correct. But what if the jaw doesn't grow properly, or if that migration is stalled for some reason?
You get low-set ears.
Exactly. And this is why low-set ears is such a huge buzzword in pediatrics and genetics. It tells you something went wrong with the migration or the growth of the facial skeleton.
So it's a red flag for certain syndromes.
It's a huge red flag. It's often a sign of chromosomal syndromes like trisomy 13 or trisomy 18, or conditions like Treacher-Collins syndrome where the facial bones just don't form properly. The ear is a bystander to the jaw's development.
And sometimes we see those little skin tags near the ear, right?
Preauricular appendages or tags, those are just leftover hillocks or accessory hillocks that didn't fuse in and smooth out. They are usually harmless, but they are a little fossil record of how the ear was assembled from 6 separate bumps.
So we have the eye and the ear both assembling at this breakneck speed in the first trimester. I want to circle back to the genetics of deafness for a second. We talked about rubella as an environmental cause, but the notes mention a specific gene, GJB2.
This is vital. We tend to think of deafness as something went wrong with the structure, like the ossicles are fused or the cochlea didn't spiral. But very often the structure looks absolutely perfect on an MRI or a CT scan.
So what's wrong?
The battery is dead.
The battery.
The inner ear relies on a very specific chemical balance, specifically potassium levels, to keep the hair cells charged so they can fire. If the potassium isn't recycled properly, The cells just go silent.
And GJB2 controls this.
GJB2 codes for a protein called connexin 26, and connexins build gap junctions, these little channels between cells. In the ear, these channels are the recycling plant for potassium.
And if GJB2 is mutated?
The recycling plant shuts down, potassium builds up where it shouldn't, the battery dies, the hair cells can't function.
Is this rare?
Not at all. This single gene mutation accounts for about 50% of all non-syndromic recessive hearing loss.
50%. That is a massive slice of the pie for one gene.
It shows how precise the chemistry of hearing really is. You can build the whole labyrinth perfectly, the loops, the spiral, the bones, but if you miss that one protein in the plumbing, you have silence.
Before we wrap up, I want to do a quick rapid-fire summary on the A conditions for the eye, just so we have the terminology locked in for our listeners who might be students. We talked about cyclopia. What about aniridia?
Aniridia. An means without, iridia means iris, so no iris. The eye looks entirely black because you were just seeing the huge, wide-open pupil. This is typically a PX6 mutation.
Vesicle.
No lens. A for without. Phakia for lens. This happens if the optic vesicle never touches the surface ectoderm properly, or the placode fails to respond. No handshake, no lens.
And entropia.
No eye at all. The optic vesicle never grew out of the brain in the first place. Or it degenerated immediately after forming.
It really underlines how every single step relies on the previous one.
It's a cascade. You break the first domino, the whole pattern just stops.
Yeah, so let's bring this all together. We started with this peppercorn-sized embryo. We watched the brain literally push out to make a camera and the skin sink in to make a labyrinth.
And we saw cartilage from the throat turn into bones that conduct sound.
It is a lot of moving parts.
It is. And what fascinates me when you look at the whole picture is the unity of it. We think of vision and hearing as totally different. Seeing is light, hearing is sound.
Different worlds.
But embryologically, they are cousins. They both rely on the surface ectoderm interacting with the underlying structures. They both rely on the same genetic toolkit, PACX genes, retinoic acid gradients. They share the same vulnerabilities to things like the rubella virus.
And they both depend on that dialogue.
That dialogue is everything. The brain asks the skin to help it see. The skin asks the cartilage to help it hear. It's a completely cooperative effort.
Here is a thought to leave you with, listeners. When you look in the mirror today, look at your pupil, that round black hole. That used to be a fissure that had to be zipped up by a microscopic biological zipper when you were the size of a bean.
And when you listen to this deep dive right now, you are using 3 tiny bones that used to be part of an ancient jaw, vibrating against a spiral tube that used to be a simple bubble of skin.
The sheer improbability that all those tissues, ectoderm, mesoderm, neural crest, all met at the exact right millisecond in the exact right place to give you the ability to perceive this moment. That is a real wonder.
It's a choreographed dance where a single misstep changes how we perceive the world forever.
Thanks for diving in with us. We'll see and hear you next time.